So I put a link in the general chat of Zulip. If you go to EuroBioC conference, everyone, underneath there's a general chat, and then it's a link to the Google Drive. So there I put a subset data that we will work on for the downstream. So you can download that. And in your local, you should do the DevTools install of this EuroBioC All-Star Workshop package. So that would install you all the packages, and then you upload this VZM HD subset data. So then we will go from chunk, let's see. Yeah, then we will progress from line 193. Is it working? Yeah, seems like mine is also working. Yeah. So has anyone also got it working? If you got it working, please raise your hand. Okay, so most of the people. Should we proceed? Do you need more time? Let me know if you need more time. Okay, so now we proceed. So yeah, you can see that under these segmented outputs, we have a H5 matrix, that is a count matrix. And then we also have the GeoJSON cell segmentation. So previously for VZM and VZM HD bind level, you have this tissue position CSV file that records X and Y coordinates. But now you have this GeoJSON file that is more like the polygon of each of the cell rather than the centroid of the cell. So you have this spatial folder, and underneath you have similar things. That's a scaling factor. That's a high or low res PNGs. And so what are we going to do is we're going to load the high res image for today, because that will help us to see the exact polygon. The low res looks really bad. I already tried. So now if you try to load the package on line 110, so you should be able to load everything. And then in order to read this function in, I actually wrote a reader myself. It's located in the utils of the vignette. Maybe I show you on GitHub. So if you go to vignettes and then here, yeah, so it's a very long journey, but I will just summarize. So what I did is to read it in as a single cell experiment. And then you will need to derive the centroid from these polygons to store it as XY, because that is needed for spatial experiment. And then I coerced a spatial experiment into the spatial feature experiment because this package, they have a good visualization, which is a package called Voyager. So it helps with visualizing polygons on top of these HNEs. But what I also did is to flip the coordinates because R and Python, I think they have a different origin set. So for R, it's on the it's on the bottom left corner, but then for Python, it's on the up left corner. So that's why we will need to flip the Y coordinates of all the polygons. So that's why I have a helper function on top. And afterwards, you would supply this polygon into the co-geometries setting of this SFE. And eventually I derived high rise in the column data so that I can help with subsetting. Here's a function for subsetting. But all of this, I submitted 2 pull requests yesterday to Visium.io and Spatial Feature Experiment. So hopefully they will merge my pull request. But for now, we can just source this script. And when you run this chunk, you should be able to read it in. No, this chunk is more to define the plotting themes. So yeah, I think I will need to navigate to the download because I installed it locally. But web one, let's see. Yeah, it's also working. So suddenly all of the data download worked. Let's see. Okay. And then yeah, so now I'm at line 165, where I define the alias for all of the plottings. It's just easier for myself to understand when I want to center a title, when I want to enlarge the legend dots. So yeah, and then afterwards, here is a repeat, but it's just to, if you have already downloaded to your cache, it won't download again, so you can safely run this chunk. And here I used my helper in the utils. I read it in as a spatial feature experiment object. It will take some time because it's a very big data. So you can see that first I would load in this GeoJSON file as a SF object. So that's why you see this shape of polygon, and you have the bounding box. Meanwhile, we're waiting for the reading of this cell segmented output. I want to show you that in the HD chapter of OSTA book, we also subset it to the same region. So exactly like you see here, the gray box is entire data coverage, and the black box is what we're going to focus on. And there's only one out of 64th region of the entire data coverage range. So it takes some effort to do the subsetting. So you see all the code here. What I did is I just recorded all the numbers, the X and Y bounding box, and then I applied it to our workshop today. It will save some time. So once it finish, I will just cut the Visium HD object to this bounding box. Yeah. So I think I used arrow, or no, maybe it's Let me see. I used stread. It's an SF package. Yeah. Yeah, it's slow. Where is it? Thank you, Martin. Yeah, so now we have it ready, so we can check what's the dimension of this object. I'll just type in the console and you can follow me, or if you already know, just do this. So now we have 118,000 genes and then 220,000 cells. So here it's just an overview of how the data looks like. These are the column datas. We have the in full res means how it aligns with the full res image, and then high res is after it times the scaling factor. And what's extra about spatial feature experiment is they have these co-geometries where it stores the centroid of the cells or the cell segmentation mask. And then here the update centroid and update cell seg is in my reader that I flipped the y coordinates of it. So later we will plot with these updated coordinates. And now we can subset it to the region of interest. So because what I subset is only a small region of it, and I know how much I want it to be reduced. So the division in terms of number of cells should be around 64 times, but it's depending on the tissue density. So for example, here it's only around 50 times, maybe because some regions are very sparse that we don't have a lot of cells. And then so here I will show you the same image, but using Voyager. So this plot image and plot image function is from the Voyager package. So if I do this, I will now have two bounding box. One is the entire data coverage. And for today, we'll only focus on this small black region. And then I will show you how the cell segmentation works over the H&E. So on the left is the H&E region zoomed into our region today. And then on the right is the segmentation bounds of all the cells. It's hard to tell, but maybe you can see here, like this black one looks like a cell, so it got segmented. And then the empty parts is filtered out due to low library size. So for example, here, I think it's, I don't know by pathology, but probably due to some folded tissue or some fibroblast that is, let's say, damaged. So we don't have any cell segmented over it. And next, we're going to talk about QC, quality check. So first, using the package Scuttle, we can derive some scuttle. We can derive some metrics, for example, the mitochondria percentage or the log library size. So here I can show you that it's now stored in the column data of this subsetted object. And then we can show them spatially. So this plot spatial feature function is also from Voyager. And here I skipped the overlaying of H&E because otherwise it would be too chaotic to see. But you can see that this is how it looks like. On the left is log library size, and on the right is percentage of mitochondria. These rings later you will see is epithelial, so that's why probably they're high in mitochondria because they're more active in their activities. So yeah, so next we are going to talk about a method that is spatially aware quality control. It's developed by the lab by Stephanie Hicks. So what they are doing is they not only filter based on library size, but they also consider the surrounding area. Is your library size an outlier compared to your neighbors? So that's making it a spatially detected outlier. So first you would run these lines that is called local outliers that will detect based on the metrics of similar to what you see in previously, but now it's computing with spatial information. Yeah, so out of all these cells, 13 are considered as outliers. And now we can see them spatially. So here, just a notation on the syntax. I find out this by ChatGPT. I find it super cool. So I have these common arguments that I don't want to repeat over the 3 lines. And for this workshop, I agreed with Ellis that we won't use lapply to plot, just for simple to illustrate. So what I do here, like the 3 exclamation mark, is calling this exact function that is from rlang package. So you put the function name here, and then all these are the like the dot 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 in when you develop a function. So here it will all these 3 lines, it will call the same common arguments. And then when you see them, it's actually very difficult to tell where is red because all the cells are so small. So you can see there are some red at the bottom here, and maybe 1 here based on low library size. And then here is 1 that's very obvious. Later we will check what this cell is exactly, but for now, remember this, we'll come back to it. So next is about cell typing. So this, it will take a while, so please let's download it. This is the Chromium reference, which is single cell reference of this Visium HD data. So it's consecutive slices from colorectal cancer, and it's from the same patient, so it should have the same cancer profile. And yeah, so we use the single cell data to do the deconvolution. That's why we need well annotated, already cell typed single cell reference. It's faster than I expected. So yeah, now it's working. And we can see that the annotated column is called level 1 here. So we have 9 cell types in total. There are B cells, endothelial, fibroblast, intestinal, epithelial, myeloid, neuronal, smooth muscle, T cell, and tumor. And we do some cleaning with the cell type names. And then because of the computational burden, we will subset per cell type to maximum 1,000 cells. So that's this line of code is doing at line 360. And then there's this cell typing method called RCTD. It's originally developed by the lab in Boston, Rafa Irizarry's lab. And it's doing, what it's doing is for spot-based Visium that they would decompose the spot into different proportions. But it can also be used for Xenium or VisGen. For example, it will just using different mode. For example, for Visium, you would use the mode of multi, and then multi or I forgot, is like the full, is full. And then for Xenium or single cell resolution, you would use the mode of doublet. So you allow at most 2 cell types per data unit. So here today we will use the level of doublet. So you can see here. And for this chunk, we commented out because it takes half an hour to run. So you can just load the intermediate data here. So now we will check, because RCTD, they also do some internal QC. Although we here say that we will filter out none of the cells, but still some are filtered out based on the gene detection rate. For example, here one cell is filtered out. So previously we have a dimension of 4,419 cells. Now we only have 4,418 cells. And the name of this cell barcode is this. So I checked what's the criteria that it got filtered out, and it's discarded due to low library size. And then I wanted to know what is the library size. So I sorted all the library size in the ascending order, and the lowest library size is 14. So this cell happened to be having the lowest library size among the entire subsetted region. So that's why it's filtered out by RCTD. And for convenience, for all the later analysis, we will just filter out this data. That's what I'm doing here. And also you can see that RCTD classified the cell typing into different class. For example, there's some cells that's rejected because it's uncertain which cell type to assign it to. And then there's doublet uncertain. That is, RCTD is confident of the first cell type, but the second one is it's ambiguous. And then doublet certain is RCTD is sure that there are two cell types, T cell and let's say B cell may be co-localized in this one cell segmentation boundary. And then most of them you can see that is actually singlet. So the data quality of this cell segmented Visium HD is not bad. So now we can visualize how it looks like. This one. So because previously there's one cell that got filtered out, so I excluded from my later analysis. So what we're doing here is to match the RCTD result into the subsetted SFE object, because later we will plot using the SFE object. So now we get to this part that we can visualize the cell types and cell type class spatially. So you can see that the yellow is singlet. Most of the cells on this part that I'm moving my mouse and here are pure cell type. And then in the middle is more like a mixture. And you can see that on top here are mostly tumor cell type. And versus on the right side here, it's epithelial. And in the middle is a lot of stroma mixed with immunes. Yeah, remember the red dot in spot sweeper that is considered a local outlier. Now we will see what's its RCTD type. So it's a doublet uncertain. So what is certain about it is the first type is T cell, but the other cell type we're not sure. So when the segmentation was running in the space ranger, maybe it included some surrounding signals. So yeah, that's why it's uncertain. Yeah. So for my own research, I usually just focus on singlets. But yeah, but I think there are definitely ways to deal with this. You can purify the uncertain ones, or you can even split the counts from the doublet certain into two cells. So there are a lot of ways you can not just throwing away your data. Yeah. So now we will filter out, we'll actually perform the QC. So previously we detect all the QC flags, but we didn't filter it out because deconvolution is usually run on raw counts and it doesn't get influenced by the low quality cells that much. And you can see there our CTD is performing internal QC as well. But for clustering, you have to be really careful. You need to make sure that your data is really filtered, QC'd, and then you run the PCA on it. So after we show you that what deconvolution can tell you about these low quality cells, and then we now filter it out from our domain identification analysis. So now we end up with a sample size of 4,406 cells. So that's what we will focus on in the downstream analysis. So now for spatial domain identification, each spatial domain can have multiple cell types in it. So that's why it's, let's say, a broader classification compared to the cell typing in the previous section. So to start with, we will log normalize our data and identify some highly variable genes. And you can also use the spatially variable genes here, but for the speed-wise, we will just use HVG. And for the method, we will use Banksy. This is a method that will incorporate a lot of, you are able to incorporate spatial information. For example, this lambda value here, you can change it. For now, I use 0.2 to use a little bit of spatial information. It's a value range between 0 and 1. So if you have time, you can change this number here and then rerun this chunk. Okay, it's running. I thought I'm running out of memory because this one's full here. So yeah, so 7 clusters are identified with the current setting. And then we put it into our column data, and then we can visualize it spatially. So here you can see that in the middle it was fibroblast, but actually the fibroblast has different spatial domain according to Banksy. And then in order to give this cluster some interpretation, we will identify marker genes from each of these domains. So here, we're computing the marker genes, and then we can visualize them into a heat map. So now what you see here is in cluster 3, these 2 genes, they are highly expressed. They're differentially expressed. And then for this cluster 6, the last 3 genes are differentially expressed. And we can also visualize these marker genes spatially. For here, I chose 3. That is MP2, PIGR here for cluster 3, and then DES for cluster 6. So if I see them spatially, it looks like this. And now what you can do is to pick a DE gene from this heat map and then plot yourself. I will also do it with you. So for example, I'm interested in PECAM1. What I'm gonna do is to have a new markdown chunk. So let's try to run this. I didn't do the palette, but you can see that PCAM1 is located in these regions. Yeah, so you can try to visualize your own gene of interest with these functions. And then now we're at this step that we want to see what's the concordance between cell typing and clustering. So let's visualize them side by side spatially first. So you can see that RCTD has a more, let's say, detailed annotation about immune cells among fibroblasts. But for Banksy here, is there are more, like, let's say, this structure identified. Maybe there are some meanings, even they are all tumor cells, majority. So that's something will be interesting to investigate. So let's see their concordance in a heat map. So you can see that cluster 4 is very likely to be tumor, and also so does cluster 7. But for cluster 2, for example, is likely to be B cell. So that's also a way to annotate your spatial domain identification. So yeah, that's all the upstream processing. Now we will transition to Ellis, who will talk more about spatial localization, neighborhood enrichment. Yeah. Have a break and then come back. That's okay. 5 minutes?
5 minutes. We start at 4 o'clock.
Okay. Yeah. Run.
Command plus. Okay. Wow, it's huge.
That's what I need.
I wish we could make this. I mean, you can always do. No, but this is not going away.
What if you wrong click and like you could probably hide that stuff?
Wait, I think it's like always show. No.
Oh, we dropped one. One's better than none. Okay.
But you could put the console here. Would that help? I usually have my console here. I don't like it down here.
Okay. How do you do that? You.
No, that's like.
I think it's in view. Sweet. Okay. Well, thank you for staying. Now we get to play a game of watch Ellis use a Mac and see how many times he presses function instead of command. I'm a Windows person, yeah. I'll hang my head in shame. I survived. Okay, so just to recap, we spent half an hour trying to download the data, and we did that. Then we walked through how to read some data in and different ways that you can QC it. So there were multiple things that would give you some sort of indication whether certain cells were measured correctly or like enough to be able to learn about them. We then, after we were kind of confident in the quantification of RNA in our different cells, we tried to use that RNA to label our cells. And so we did that labeling in two ways. One, we used a deconvolution approach to use a single cell reference to project those labels onto our data. And the other, we used an unsupervised clustering technique to group cells into cells that have similar kind of features and then potentially the features around them. I think Banksy uses as well. So that's where we're at. And so now we're going to try to move on and start asking some questions with our data. And so we could obviously sit here and just think of random questions, but I think it's always important to put experiments back in context. And so in my brain, there are two key reasons. Let's say there's three key reasons that someone would do a spatial omics experiment. The first key reason is that they hear that everybody else is doing it, and so they feel like they must do it too. The second reason is because they've been working with a tissue that is potentially a little bit fragile, or they're interested in cell types that are difficult to measure with other technologies, and they see a spatial technology as a way to potentially measure these cell types that don't make it through flow cytometry or single cell sequencing. And so as it stands, if I was one of those people, I have now got my data in a position where I can learn something. So if we scroll, like even just to this plot, I've got a situation where now I've measured lots of cells and I've labeled them all. And if I simply wanted to know which cells were in this tissue, I'm now in a position to say that. And so that might sound a bit obvious, but I work with lots of people where just being able to say which cells are in my tissue is their research question. The third question, or the third reason you would do some sort of spatial omics experiment is because you're hoping to learn something about the spatial arrangement of your cells. And so now we're going to talk about different ways we can learn about spatial arrangements. And so I would like to ask you, what do you think the first thing you should do when you're trying to learn something about the spatial arrangement of your cells? Pardon? Distance. So you could calculate distances, or when in doubt, look at your data is generally the kind of catchphrase. And so we've got this great spatial kind of computational thing in our brain. Our eyes are really good at seeing spatial patterns. If you want to see if there's any sort of spatial relationship in your images, the first thing you should do is look at some of your images. And so if we were to look at our images, at least using this visualization, we can see that it's highly structured. So we know that we've identified these tumor-rich regions. We know that we've got these epithelial regions. What are they called? Ducts, or does anyone do colon? Crypts. And so they're very homogeneous regions. They're full of the same cell type. We've also got this big stromal region in the middle that's a big mismatch of everything. We can see that we've got these smooth muscle, kind of thin sliver. Just looking at this image, we start to get a pretty rich understanding of how our tissue is organized. But if we had lots of images, it becomes hard to look at lots of images. And so that's where quantifications can start to help. Or potentially, like we've got lots of we've only got 9 cell types here. And this is starting to get pretty complex to look at. In that RNA-seq data set that we used as our reference, it had our level 1 clustering, which has the 9 different cell types. It's also got a level 2 clustering, which has a lot more than 9 cell types. Let's say 26 cell types or something. So if you were to try to plot 26 different colors on this plot, it would very quickly become overwhelming. So now we're going to, with the next 20 minutes, go through and look at the some different types of questions you can ask by quantifying spatial relationships. So the first thing we're going to start by doing is we're going to use the K-cross function, which Martin talked about an hour ago, to quantify how close different cell types are, how localized they are, or how much they attract to each other or avoid each other. And so the K-cross function, effectively just for every single cell type, you calculate the proportion of another cell type within a radius around it. And so I'm going to use the get pairwise function from the SpicyR package. And it's got lots of different arguments that you can give it, but here we've given it our spatial feature experiment. Got that round the right way? Great, thumbs up. I'm going to give it the radius of that I want to calculate different cells near it. And so here I've set 200 pixels, which is about 50 microns. I've told it what my cell type column is, what my sample ID column is, and in this case, we've only got one image, so that's great. And also the names of my spatial coordinates. We're going to run that, and that shouldn't take too long. Oh, look, Mac scrolls the opposite direction. Great. So this has gone, and it has calculated. Oh, no. I'm going to need you just to stand next to me, Eleanor. There we go. I'm amazing. Cross K. What does this look like? So this doesn't look pretty, but it's just a vector, and it tells you smooth muscles, what is the relationship between smooth muscles with other smooth muscles? And it gives us a value of 364. It's positive, which means they're attracted. In terms of the scale of that, who knows what it means? Then if you were to look at neuronal cells and smooth muscle cells, it's negative 56. So it's negative, it means they're avoiding each other. Who knows what the 56 means? But 56 is less than 300, so that's some sort of scale, right? But just looking at this, it's like you could scroll through these results. They probably don't make much sense. So it's worth looking at a visualization just to rapidly absorb some information. And so here we have a plot. And so each of these circles is the spatial relationship between two different cell types. If it's red, it means they're attracting. If it's blue, it means they're avoiding. And so I would like you to look at this plot and think, hey, what sticks out to me? Like what's interesting? What kind of patterns can I see? And so when I look at this, I see that neuronal cells seem to be highly attracted to neuronal cells. And so if you wanted to scroll up, I hate Max. We can see that the neuronal cells with this thin little strip here. So in terms of the image space, they're all localized into one little strip of the image. So of course, they're like highly localized with each other. We can see smooth muscle cells are attracted to each other. But otherwise, there's two other clear patterns that I see, which is that tumor cells are avoiding all other cell types apart from themselves. And that makes sense when you look at the actual image, right? There was that big compartmentalized bit of tumor and then everything else. So everything else is avoiding tumor. We also see that everything else is pretty much avoiding the crypts, those epithelial cells. And again, if you remember back, they're really highly compartmentalized. And then because those two things, because all the other cell types are avoiding those two big compartmentalized regions, everything else seems to be attracted to each other. And so if I was interested in cancer, I think a reasonable thing to ask would be, okay, what cells are hanging out with tumors? What's attracted to tumor? And just looking at this plot, looking at this quantification, you would say, oh, nothing is. And so it's like, okay, well, what did I learn? Not too much. So this is our first spatial quantification, but it's definitely not where we need to stop. If I really want to know what is interacting with tumor, how can I start to reframe my question so that I might learn something about that? So, what's this generate? Oh, that just generates that plot all over again. We'll use it in a sec. Okay, so now we are going to use the k-cross function in a slightly different way. We're going to slightly tweak the question that we're asking. And so we've got a package called Spatial, and it's got in that it's got a function called contextual. And what contextual allows you to ask is, instead of just asking, are two things close to each other or two things far away from each other, we use contexts to define our question. So we might like to ask, are T cells close to tumor cells, given where all the other stromal cells are in the tissue? So relative to all the other cells, are T cells actually just a bit closer? Does that kind of make sense? And so by acknowledging that there is structure in my data and acknowledging that structure, we can frame our test a slightly bit different. And so here, we're going to use our immune cells as a parent population or as our context, and ask the question, are any of these three immune cells localized with tumor relative to those three immune cells? So here I've set my parent population as immune. When you run contextual, you need to run this parent combinations function, which will go and structure things in different ways so that the function can work. And so you've got to tell it what all the different cell types you want to analyze are. And then you can just list multiple parent populations. So in this case, we're just giving it a single parent population, but you could give it multiple parent populations or multiple contexts, and we'll analyze them all simultaneously. And then much like SpicyR, we can run this, we give it our data, we give it our parent information, tell it where some of our columns are, and we're setting a radius of 200. Why are you setting a radius of 200, Ellis? How do you know what radius to choose? There is no right answer. You just use your gut, and if you're looking for cells that are really close to each other, use a small radius. If you're looking for relationships that might be a bit broader, use a bigger radius. Or if you're really, really intelligent, you talk to Martin, and you analyze all radiuses simultaneously and do some functional data analysis. Okay, so what does this give us? So this will give us a big matrix of results, which I was dumb enough to print out. And so here we've got T cells. So in the context of where immune cells are, T cells and B cells would be near each other if you use just a normal K-cross function, or they would be avoiding each other if you use this context. So if you looked at immune cell, like just where the immune cells are, they're actually avoiding each other. But when you kind of put in the context of the whole image, they look like they're close to each other because they're all in the same little compartment. Right. So we've done these two different tests. It's probably worth looking to see the relationships between these two different tests. So here, all I've done is plot a standard K-function test with our contextual test. And we can see that they're reasonably correlated. So if something was so if neuronal cells were attracted to B cells in both the K function and also in contextual, epithelial cells and T cells were both avoiding each other, whether you use contextual or the K function. But interestingly, up here, we have this nice little relationship where in the original K function, tumor cells and myeloid cells were avoiding each other. But then once you acknowledged in the image where immune cells are, myeloid cells now look like they're actually closer to tumor cells. And so we could kind of get a gut feeling from that a little bit from our original tests. So if we went back and looked at this little bubble plot, we can see that out of all the cell types, myeloid cells are the least avoiding with tumor. So that's kind of makes some sort of intuitive sense. But if we actually go and look where these myeloid cells are in the image, oh, we don't need to do this. So this is just to make Martin happy. And so we could look at these K functions and contextual functions over all radiuses just to kind of get a feel for how things are related. So we can see that if we had chosen a really small radius, we wouldn't have seen really any relationship between tumor cells and myeloid cells. But as our radius expands, the relationships become stronger. And here it looks like it has at least plateaued. If not, it might drop down again afterwards. But again, contextual shows a positive relationship and just a K function shows a negative relationship. All right, so like I said, let's go look at where these tumor cells are and the myeloid cells. And so here I've plotted myeloid cells in red, tumor cells in blue, and then the other immune cells are in gray. And so I don't know how great your eyes are, but when I squint at this, what I see is that potentially like there are a lot of red cells near tumor, at least on the border. Down here, you've got a lot more gray than you have red. So maybe red is a bit more attracted. But in particular, you have these myeloid cells infiltrating the tumor, and there's no other immune cell infiltrating the tumor. So we don't have our infiltrating T cells, we don't have B cells, we have myeloid cells. And so this is one of the big things that is driving this relationship. The myeloid cells are infiltrating the tumor. So I reckon that's a pretty neat thing that we found. And if we had just looked at the image with our eyes, I don't think we would have necessarily seen that relationship, and I don't think it would have stood out as something interesting. So a quantification has helped us learn something. All right, so we've talked about the spatial relationship of cell types, just as cell types alone. Now I want to at least pose another question. So if we know that these myeloid cells are infiltrating the tumor, is the gene expression of the myeloid cells infiltrating the tumor any different to the gene expression outside the tumor? So if myeloid cells have a lot of tumor cells near them, do they have different gene expression to those that don't have lots of tumor cells near them? And so again, we've got a function called spatiomark. which will do something really, really simple. So it'll go through and it'll calculate the number of a particular cell type near a particular cell type. So here I've used the get abundances function with a radius of 200 again, and for every single cell calculates the number of cells in that radius, the number of the different cell types in that radius. We've then got this function called calc state changes, which effectively just goes and does a regression, plotting the abundance against the expression for every single gene. So it's a really, really simple test, right? You've got the abundance, you've got the expression, and then depending on which way the stage is pointing, the gene expression might go up as the abundances get small, or gene expression might go up as the abundances get big. So we're going to run this specifically looking at a relationship between myeloid cells and tumor cells. Because we have count data, I've also used this test equals negative binomial, which effectively just leverages edgeR to go and do our tests. So we're going to assume that we've got Poissonish data. And so what do we find? What we find is that we've got all of these genes, and IGHG3 seems to be going down in expression as myeloid cells get close to tumor cells. ChatGPT says this gene shouldn't even be expressed. I think it's mostly expressed in B cells, and so this, in a weird way, makes sense. It's like, well, when myeloid cells are where B cells are, which is where tumor cells are not, they're being contaminated by a B cell marker. But when they go into the tumor where there's no B cells, that marker is no longer expressed. So it's not like hard biology, but it's technical artifact that's representative of the biology we're measuring. The next gene is SPP1, and I know from some of my melanoma work, this is a known gene that's associated with tumor, what's the word they use? TAMs. Tumor-associated macrophages. Okay, so let's look, because we're running out of time, let's look at SPP1 in our image. And so we could go look using the plotting functions we did before, but instead we're going to use this plot state changes because it allows us to do a few things. So in this plot, we have two or three things highlighted. So the density kind of pattern that you can see is the density of tumor cells. They're also, all those tumor cells are also plotted in these small blue dots. Then all of the slightly bigger dots are our myeloid cells. And if you've got brilliant eyesight, you should be able to see that the myeloid cells that are infiltrating the tumor, at least some of them have got pretty high SPP1 expression. And so just to show you something as well, to illustrate the test that we're doing, this is the relationship that we fit. So each of these dots is a myeloid cell. And on the x-axis, we have the number of tumor cells near that myeloid cell. And on the y-axis, we've got the expression of SPP1. We can see that there were very, very few cells actually expressing SSP1. But if you don't have any tumor cells near you, you are not expressing SPP1. And if you do, you've got a tiny chance that you might. Right. So another thing to notice is if we flick through our results, not that way. So again, I'm not great with gene names. I'm not a biologist who can look at genes and just make a story. But one thing that did stick out to me with this was this KIRCAM6, which I know is not a gene expressed in myeloid cells. It is a gene typically expressed in like fibroblasts and tumor cells. And so what does it mean if we're seeing like a tumor gene being expressed in myeloid cells when they're near tumors? It probably means, again, that we've got this kind of technical artifact happening in our data. Like we saw the segmentation that we're trying to do. Sometimes when you segment, you end up getting multiple cells in the same cell. So maybe we're segmenting 2 cells at once, or most of these RNA technologies, when they kind of like squish stuff when they're doing their processing, RNA often diffuses. And so it kind of moves outside the cell body into other arguable cell bodies. And so you get this kind of spillover. And so typically when I do this type of analysis, the majority of results that majority of genes that I find are all driven by spillover, and you've kind of got to sift through and find the ones that actually biologically make sense. We were a little bit frustrated with this, and so we tried really hard to think of a way for, actually this was just a proof of concept. If we go back and look at our single cell data, it must be a big plot, we'll end up seeing that KIACAM6 is expressed in the tumor cells. So we came up with this method for trying to account for the fact that some cells will be contaminated by other cells. And so in StatioMark, we've got a function that tries to estimate these contamination factors. But one of the great things about using RCTD to do our cell type annotation is because it's doing deconvolution, it's effectively giving us a measure of how contaminated each cell is. And so I don't need to go and calculate a contamination score. I can just use the ones that were given to us by RCDT. So let's look at what that looks like. So out of RCTD, for every cell, they give you a score of how likely that cell is to be another cell. And so if we look at cell 3, it was 0.69 of an endothelial cell, or 0.3 of a fibroblast, and then kind of 0 of everything else. And so this tells us that cell has some endothelial genes and some fibroblast genes. It's probably a mix of both. There's 2 cells sitting next to each other. It's ambiguous. It's a doublet. Anyway, we can dump these into a reduced dimension in our spatial feature experiment and then pass that to our calc state changes again. And then, so now when we do that, if we go and look for our KIACAM6, I am standing up, watch. They wanted me to get moving. I'm sorry I'm stationary. My heart's going to explode. So when we run this, it's going to go and calculate these relationships. And what we're going to see is that KIACAM6 is no longer near the top of our list. It's still got a small p-value, so we didn't do a great job of correcting for things. But in this particular case, it comes nowhere near like our FDR threshold. And so it's like fallen out of the top things. So let's look at that. We've got the expression of KIACAM6 in myeloid cells as they get close to tumor cells. It's a positive coefficient, so it's going up as more tumor cells are near. The t-value is 2.4, which if we know our rule of thumb, if the t statistic is greater than 2, it's likely to have a p-value less than 0.05. And lo and behold, it does. The p-value is around 0.01, which is not strong enough to overcome any threshold. So it's dumped down in the list. So by taking into account our RCTD deconvolution results, we can kind of hand wave and adjust these spatial tests. So if we had more time, I'd be telling you to do one of 2 things. And so one of those would be to go use the level 2 annotation of the cell types and look to see what relationships you could find in the data there, particularly going back to that contextual function and defining different parent populations. So you might define like immune cells, or you might say, I'm going to look at all the different T cells populations, and you could play around there. Another thing that you could do is, while RCTD is a very well-performing deconvolution method, it is very, very slow. CARD is very, very fast, but it's arguably, at least in benchmarking studies, it hasn't been performed quite as well. And so you could run this instead, and see how that goes and affects your gene expression results with spatial microcontrollers. But that's where I wanted to leave it. I'm sorry that you didn't get an opportunity to go and explore things a little bit more yourself, but hopefully you learned something by just having me stand here talking at you. So we have hit 4:31, and so we're officially behind schedule. I know a lot of you are presenting posters, so I'll leave you quickly. But I do want to thank you for coming and attending our workshop. I hope you got something out of it. Then I'd also like to thank Estella for putting in a ton of work pulling this thing together. There was a lot of back and forth trying to get things to compile. Things are never clean and easy. And reading in this data was non-trivial. And so I'm really looking forward to your pushes to some of these packages so we get these standard reading functions. And then of course, I'd like to roll back right to the beginning. If I knew how to use a Mac, I would put the OSTA book back up and go to the acknowledgement slides and acknowledge all of the people that have been contributing to these work. So for a second, you could just imagine that I'm Ellis. This is me being Ellis, and you can clap me as I list. So thank you again, and I look forward to chatting to you all over the next 24 hours. I don't know how to.
Okay, so this finishes this